Every 2 minutes, someone in England gets that diagnosis cancer. And for about half of us born since 1960, it's something we might face ourselves. It sounds pretty daunting, doesn't it? But understanding why and how it happens, well, that's the first really powerful step, especially for you, our future medical professionals. So today we're doing a deep dive specifically for you into the really intricate world of carcinogenesis, basically how a normal cell transforms into what we call a neoplasm. Now, neoplasm, it sounds a bit clinical, maybe even scary, but let's break it down. Neo just means new, plasm means formed material. So really, it's just a new collection of cells, cells that have started multiplying abnormally, and crucially, they keep doing it even after whatever started it is gone.
Exactly. And it's so important to grasp right from the start that not all neoplasms are malignant, what people generally mean by cancer.
Hmm.
We obviously worry most about the malignant ones because they can invade and spread, but benign neoplasms, they're also really significant medically. Think about uterine fibroids. Super common, maybe 80–90% of women get them by age 50. Benign, yes, but they can cause real problems, pain, fertility issues, or meningiomas, brain tumors, making up over a third of primary CNS tumors. Often benign, but their location alone can cause serious neurological deficits. So getting the mechanisms behind both benign and malignant growth, that's absolutely foundational for you.
Okay, so our mission today is cracking how these cells go rogue. But the big question, the one for you to keep turning over in your mind as we go, is why? What are the fundamental things pushing a cell off its normal path towards this uncontrolled growth? Keep that question handy.
And to help navigate this complex area, let's use a kind of mental map. A framework, we like to call it CRABS just to make it easier to remember, stands for the 5 key landmarks we'll hit: chemicals, radiation, agents like infections, breakdown in genetic control, and finally, steps in progression. We'll unpack each one.
Right, CRABS. Got it. But before we start that journey, maybe let's just put some numbers on this, get a sense of the scale. Globally, it's huge. 82 million new cases back in 2020. And things like lung, breast, bowel, prostate cancer, they make up what, over 40% of that? Here in the UK, about 385,000 new diagnoses in 2019. But there's actually some really good news mixed in there. Survival rates. They're the highest they've ever been. More than half of people diagnosed now live 10 years or more. That's real progress.
It absolutely is. And what's really insightful, I think, beyond survival is the preventability. Around 38% of cancers are thought to be preventable. And if you look at cancer deaths, about 30% are linked to just 5 behavioral and dietary things: high BMI, not enough fruit and veg, inactivity, smoking, and alcohol. Tobacco alone, that's maybe a quarter of all cancer deaths. It really drives home the massive impact of these modifiable extrinsic factors, things we can actually change.
That's a really powerful point. Modifiable risk. So the cause isn't just one thing, is it? It's this complex dance, like you said, multifactorial, between intrinsic stuff inside us and extrinsic stuff from the outside. I like thinking of it like a plant. The intrinsic factors are like the seed's genetics, its built-in traits, and the extrinsic factors, that's the soil, the sun, the pests, the water, its whole environment.
That's a great analogy. So for us, intrinsic factors are things like heredity, genes we inherit. Our age being over 65 is a big risk factor for many cancers and gender, often due to hormones.
Yeah.
These are mostly things we can't really alter.
Right, but then the extrinsic factors, these are environmental exposures, lifestyle choices. And you said these account for, what, 85% of a population's risk?
Roughly, yes. And the key is many are modifiable. Things like diet, smoking, sun exposure, even broader things like socioeconomic factors and access to healthcare play a modifiable role here. They affect exposure and outcomes.
It really shows how our cells are kind of caught between our internal makeup and the world hitting them. Okay, let's start the CRABS journey then. First up, C for chemicals, R for radiation, A for agents, the external triggers.
Okay, chemicals. These teach us a lot. Many chemicals enter our bodies actually pretty harmlessly. They're pro-carcinogens, like unlit fireworks, as you said earlier. They need something to light the fuse, usually an enzyme, often one from the cytochrome P450 family. These are really important enzymes, especially in the liver, involved in metabolizing drugs and, well, activating these chemicals. Take benzopyrene from cigarette smoke. It's a procarcinogen. But enzymes like CYP1A1 convert it into benzopyrene diallipoxide. That's the active form. That active form is what sticks to DNA, causing mutations.
Ah, so that's bioactivation, our own body's machinery accidentally weaponizing these compounds. Okay, can you give us some key examples? How do they work?
Sure. Let's look at a few major players. Polycyclic aromatic hydrocarbons, PAHs, found in tar, mineral oil, smoke. Like benzopyrene, they get activated to epoxides, bind to DNA, cause mutations. Lungs and skin are common targets. Then aromatic amines, like 2-naphthylamine, used historically in rubber making dyes. The interesting thing here is how it hits the bladder specifically. It gets processed in the liver, made safe by adding glucuronic acid. But then, in the kidney and bladder, enzymes remove that protective group, exposing the bladder lining directly to the carcinogen. Nasty stuff.
Wow, that's incredibly specific targeting by the body's own processes.
It is. Then you have nitrosamines from nitrites in processed foods or fertilizers. Gut bacteria can convert these into carcinogens that damage DNA, often linked to gut cancers. And finally, asbestos. Those silicate fibers used in building materials now banned, thankfully. It's more about physical damage to the lung tissue. The really striking thing with asbestos is the latency, often 30 years or more, between exposure and getting malignant mesothelioma or lung cancer.
30 years? That long delay and the organ specificity you mentioned with aromatic amines. It really suggests this isn't usually a quick single event, does it?
Exactly. It points towards a cumulative process, which fits perfectly with the steps and progression we'll discuss later under S. But hold that thought. Let's move to R, radiation. This is about energy hitting our cells. UV light from the sun, we all know about skin damage, but ionizing radiation X-rays, nuclear radiation, alpha, beta, gamma rays, that's powerful enough to knock electrons off atoms. This causes direct DNA damage, breaks in the DNA strands, changes to the bases, but it also works indirectly by creating free radicals inside the cell. These free radicals are highly reactive molecules and they just wreak havoc, damaging DNA and other cell parts. Now, our cells have repair crews, DNA repair proteins trying to fix this, but sometimes they miss things or they make mistakes during the repair, misrepairs, and those mutations stick.
And that's where conditions like XP come in, xeroderma pigmentosum.
Precisely. People with XP have faulty DNA repair genes, specifically for damage caused by UV light. Their repair crew for UV damage is defective, so they're incredibly sensitive to sunlight and get skin cancers very young. It highlights how crucial that repair process is.
Okay, so chemicals, radiation. What's the last external factor? A for infectious agents. Bugs causing cancer. How does that work?
It's a really important area, and these agents can work in two main ways. Directly or indirectly. Direct agents, like certain types of human papillomavirus, HPV 16 and 18, are quite clever in a sinister way. They actually integrate their own viral DNA into our cell's genome. Then they express viral proteins, specifically E6 and E7. E6 targets our P53 protein for destruction, and E7 inhibits our PRB protein.
P53 and TRB, wait, aren't those the really important break proteins we'll talk about later, the tumor suppressors?
Exactly. So HPV essentially disables two major breaks on cell growth. This directly pushes the cell to keep dividing and stops it from undergoing apoptosis, or programmed cell death. That's the link to cervical, vaginal, penile cancers. Then you have the indirect agents. They don't directly mess with the growth genes like HPV does. Instead, they cause chronic inflammation and tissue damage. This constant injury forces the tissue to regenerate, meaning cells are dividing more often than usual. This ongoing cell turnover acts as a promoter.
A promoter? Like encouraging cells that already have some damage to multiply.
That's right. It increases the chance of errors during DNA replication, and it helps any already initiated cells with that first hit to expand into a larger population. Think of hepatitis B or C viruses causing chronic liver inflammation, leading to liver cancer, or Helicobacter pylori bacteria causing chronic gastritis, increasing risk for stomach cancer. It's the chronic inflammation driving the process.
That makes sense. So constant repair and regrowth creates more opportunities for things to go wrong. Are there other examples?
Oh yes. Epstein-Barr virus, EBV, is linked to certain lymphomas and nasopharyngeal cancer. HIV doesn't directly cause cancer, but by weakening the immune system, it allows other viruses like HHV8 to cause Kaposi sarcoma. And even fungi can play a role. Aspergillus flavus, a mold that grows on crops like peanuts, produces aflatoxin B1. It's metabolized in the liver to a potent carcinogen linked to liver cancer.
Wow. Chemicals, radiation, infections, the external threats are incredibly diverse.
Oh, yeah.
Okay, so that covers the outside forces. What about the battle inside the cell? That takes us to B in CRABS, right? Breakdown in genetic control.
Exactly. This is about the cell's own operating system going haywire. But first, a really neat piece of biology that tells us something fundamental. How do we know cancers usually start from just one single rogue cell?
Yeah, how do we know it's not like a whole group of cells deciding to rebel at once?
It comes down to studies in women looking at genes on the X chromosome. There's a process called lionization, or X inactivation, where in each female cell, one of the two X chromosomes is randomly switched off early in development. So if a woman is heterozygous for a gene on the X chromosome, like the one for the enzyme G6PD, her normal tissues will be a mosaic a mix of cells expressing one version of the enzyme or the other.
Okay, a patchwork quilt of cells.
Precisely. But if you look at a tumor from that same woman, you find that all the tumor cells express only one version of G6PD, not the mix.
Ah, so they must have all come from a single original cell that had already undergone X inactivation. That's clever, monoclonal origin.
Exactly. It proves the tumor started from one ancestor cell,
Yeah.
which makes understanding the genetic errors in that first cell even more critical.
Right. So let's talk about those errors. You mentioned the cell's operating system. What are the key parts and how do they break down? You used a car analogy earlier.
Yes, the car analogy works quite well. Think of cell growth control like driving a car. You need accelerators, brakes, and mechanics to keep it running right. Cancer messes with all three. First, the accelerators. Proto-oncogenes. These are normal genes. When they get the right signal, they promote growth and division. Perfectly normal, necessary function. But if they get a specific type of mutation, a function-altering one, they become oncogenes. Now it's like the accelerator pedal is stuck down. Constant growth signal.
Always accelerating even when it shouldn't be.
Exactly. And here's a key point. You usually only need one copy of the proto-oncogene to mutate into an oncogene to get this effect. One hit is enough.
Examples.
Classic examples are RES genes. They're involved in signal transduction, kind of relaying the grow message. They're mutated in maybe a third of all human cancers. Also MYC, a gene controlling transcription, and HER2, a growth factor receptor often amplified in breast cancer.
Okay, so oncogenes are jammed accelerators, one hit needed. What about the brakes? Are they different?
They are different, and crucially so. These are the tumor suppressor genes, TSGs. They are the brakes. They normally inhibit growth, trigger DNA repair, or tell a badly damaged cell to undergo apoptosis self-destruct for the greater good. They're gatekeepers.
So if they break, the car can't stop.
Right. But unlike oncogenes, for the brakes to fail completely, you usually need to lose the function of both copies of the TSG. Remember, we inherit 2 copies of most genes, one from each parent.
Ah, so you need 2 hits to knock out the brakes, like needing both front and back brakes to fail.
That's a good way to think of it. One working copy is often enough to do the job. This is called haplosufficiency. You need to lose both.
Key examples of these TSGs.
The absolute classics are PRB, the retinoblastoma protein. It controls a critical checkpoint in the cell cycle. Lose it, and cells divide when they shouldn't. And then there's P53, often called the guardian of the genome. It senses DNA damage and can halt the cell cycle for repair or trigger apoptosis. It's mutated in over half of all human cancers. Losing P53 is a huge step towards malignancy.
Accelerators, brakes. What about the mechanics?
Ah, yes, the caretaker genes. These are the mechanics responsible for fixing DNA damage, maintaining genomic stability. They don't directly control growth like oncogenes or TSGs. But if your caretaker genes are mutated, the mechanics are on strike. DNA damage doesn't get repaired properly. This leads to a much higher mutation rate, overall genomic instability. Small errors accumulate much faster.
So faults in the mechanics make the whole car more likely to break down in other ways.
Exactly. Prime examples are BRCA1 and BRCA2. They're involved in repairing double-strand DNA breaks. Mutations in these dramatically increase the risk of breast, ovarian, prostate, and pancreatic cancers. And the XP genes we mentioned earlier involved in fixing UV damage. They're also caretaker genes.
This idea of needing 2 hits for the tumor suppressor genes, the brakes, you said it came from studying retinoblastoma, that eye cancer in kids.
Yes, a really seminal piece of work by Alfred Knudsen back in 1971. He looked at this rare childhood eye cancer. He noticed 2 patterns. Some cases were familial, ran in families, often affected both eyes and appeared early. Others were sporadic, no family history, usually just one eye, and appeared later.
Okay.
Knudsen proposed the 2-hit hypothesis. He said you need 2 mutational events to knock out the RB1 gene, a TSG, to get the cancer. In the familial cases, the child inherits the first hit in the RB1 gene through the germline. So every single cell in their body already has one faulty copy. They only need one more spontaneous somatic mutation, the second hit, in a retinal cell. Much higher probability, hence earlier onset, often bilateral.
Right, they start halfway there, basically.
Exactly. But in the sporadic cases, the child starts with 2 perfectly normal RB1 genes, so that one unlucky retinal cell needs to suffer 2 independent somatic mutations, hitting both copies of RB1. Much rarer event, hence later onset, usually unilateral.
That makes perfect sense. It elegantly explains the difference and highlights the importance of those 2 hits for TSGs.
Okay, so we've got the external triggers, C, R, A, and the internal genetic breakdown, B. Now, thinking back to that long latency period for things like asbestos, that brings us to S, right? The steps in progression.
Precisely. Cancer doesn't usually happen overnight. It's a multi-step journey, often taking years, even decades. We can generally break it down into three key stages: initiation, promotion, and progression, or IPP.
Initiation, promotion, progression. Okay, what's initiation?
Initiation is the first hit. It's an initial irreversible genetic change, a mutation in a single cell's DNA, often caused by one of those chemical or radiation initiators we talked about. Think of it like making a tiny, permanent crack in one brick in a very large wall. By itself, maybe not a big deal yet.
Okay, one cracked brick, then promotion. Promotion is where that initiated cell, the one with the cracked brick, starts to multiply. Promoters, things like chronic inflammation, certain hormones, even some chemicals that aren't mutagenic themselves, they don't necessarily cause new mutations. Instead, they create conditions that encourage that one initiated cell to divide and divide, forming a clone, a whole population of cells carrying that initial mutation.
So promotion is like selectively encouraging the growth of the damaged bricks, making a bigger patch of weakness.
Exactly. It expands the population of initiated cells, making it much more likely that one of them will eventually suffer the next critical mutation. It's often reversible if the promoter is removed.
And finally, progression.
Progression is where things really accelerate towards malignancy. Within that growing clone of initiated and promoted cells, additional mutations start to accumulate over time. These hits affect more oncogenes, more tumor suppressor genes, more caretaker genes. The cells become more genetically unstable, they grow even more uncontrollably, they start to look more abnormal. Eventually, they acquire the ability to invade surrounding tissues and metastasize spread to distant sites. This is the stage where the bad situation gets progressively worse, leading to a fully malignant tumor.
So it's a snowball effect. One mutation, then proliferation, then more mutations building on each other. Is there a classic example of this?
Absolutely. Colorectal cancer is the textbook example. It often follows what's called the adenoma-carcinoma sequence. It starts as the benign polyp and adenoma. Over years, even decades, cells within that polyp acquire a series of specific mutations, often starting with the APC gene, a TSG, then maybe KRAS, an oncogene, then P53, another TSG. Each step makes the polyp grow larger, more abnormal, until eventually it becomes an invasive carcinoma. It beautifully illustrates that stepwise progression.
Okay, so after all these steps, after accumulating all these changes, the cell becomes fully malignant. What defines that state? What are the common characteristics?
Right, so researchers have identified a set of core capabilities that most, if not all, malignant cancers acquire during their development. We call them the hallmarks of cancer. They're the defining traits. For remembering them, especially for exams, the mnemonic Grimes plus die can be helpful. It covers the main ones.
Grimes plus die. Okay, let's
Right.
break that down. G.
G is for self-sufficiency in growth signals. Cancer cells don't wait for external go signals. They generate their own or have pathways stuck in the on position, like a car with the accelerator jammed. R is resistance to growth stop signals. They ignore the signals that normally tell cells to stop dividing, like those from TSGs like PRB and P53. Ignoring the red lights. I is mortalization. Normal cells can only divide a limited number of times. Cancer cells often reactivate an enzyme called telomerase, which lets them divide indefinitely, never running out of fuel. M is metastasis. The ability to invade local tissues and spread to distant parts of the body, leaving the road entirely. This is often the deadliest hallmark.
E.
E is evading the immune system. Normally, our immune system can recognize and destroy abnormal cells. Cancer cells develop tricks to hide from or suppress the immune response. Cloaking from the police.
Us.
S is sustained angiogenesis. Tumors need nutrients and oxygen to grow beyond a tiny size. They trick the body into growing new blood vessels to supply them,
Oh,
building their own fuel lines.
okay, that's Grimes. What about die?
Right, these are slightly newer additions, often seen as enabling characteristics. D is for deregulated metabolism. Cancer cells often rewire their metabolism, like favoring inefficient energy production, glycolysis, even when oxygen is present, the Warburg effect. This helps fuel rapid growth. E is genomic instability. This relates back to those faulty caretaker genes. The underlying instability allows mutations to accumulate faster, accelerating the acquisition of the other hallmarks, the car constantly breaking down and poorly rebuilding itself. And E is tumor-promoting inflammation. We touched on this with indirect infectious agents. Inflammation in the tumor microenvironment can actually supply growth factors, help with angiogenesis, and promote invasion. A fire under the hood, helping it run.
Wow. So acquiring these hallmarks is the end game of carcinogenesis. And going back to our very first question, why do cells go rogue? The answer seems to be this combination of external hits and internal genetic failures that allow cells to tick off these boxes, right? To achieve this uncontrolled invasive growth.
That's the essence of it. It's that interplay. And we see evidence of this interplay in things like geographical variations in cancer rates.
Hmm.
Cancers like stomach cancer, breast cancer, Burkitt's lymphoma, their incidence varies hugely around the world. Migration studies are fascinating here. When people move from a high-risk area to a low-risk area, their cancer rates, or those of their children, often shift towards the rates of the new home country. It really points to the power of those extrinsic environmental factors.
That's really compelling, and it ties back to what you said about modifiable risks, things like diet, lifestyle, exposures, specific occupations too, right? Asbestos workers, dye workers.
Absolutely. Clear links there. Which brings up another related question, one I posed earlier for you listeners to think about. What about pre-existing medical conditions? We know things like ulcerative colitis raise colorectal cancer risk, cirrhosis raises liver cancer risk, Hashimoto's thyroiditis raises thyroid cancer risk, chronic gastritis raises stomach cancer risk. What's the common thread, particularly with these inflammatory conditions? Why do they consistently increase cancer risk? We'll circle back to the answer right at the end.
Okay, a cliffhanger. But given all this complexity, this multi-step process, this interplay of factors, it really underscores why early detection is so vital. That brings us to screening.
It does. Screening programs are our best bet for catching cancers early, often before they've even caused symptoms, when treatment is generally much more effective and survival chances are highest.
But setting up a good screening program isn't simple, is it? There are criteria.
Definitely. We often refer to Wilson's criteria, developed by the WHO. For a screening program to be worthwhile, the condition needs to be a significant health problem. We need to understand its natural history, how it progresses, there needs to be an effective treatment available, and the test itself. It has to be relatively simple, safe, accurate, reliable, and acceptable to the people you're asking to take it. Plus, you need clear policies on what to do with positive results and how to manage the costs. It's quite a checklist.
Makes sense. So what are the main established programs we have here in the UK?
Well, the big three national programs are: for cervical cancer, women aged 25-64 are invited for screening, initially using cytology, a smear test, but increasingly moving towards primary HPV testing given HPV's direct role. For breast cancer, women aged currently 47-73 are invited for mammograms every 3 years. And for bowel cancer, men and women aged 60-74 get a home testing kit called FIT, fecal immunochemical test, to check for hidden blood in stool. And there's also a one-off flexible sigmoidoscopy offered around age 55 in England.
So those are key defenses based on understanding the risks and progression we've been talking about. Okay, let's try and bring this all together.
Right. So we've journeyed through that CRABS framework today. We looked at chemicals and radiation as external initiators. We saw how infectious agents can work directly or indirectly. We delved into the breakdown in genetic control, those oncogenes, accelerators, tumor suppressors, brakes, and caretakers, mechanics, and the two-hit idea. And finally, the steps of progression, initiation, promotion, progression leading to the hallmarks of cancer. Hopefully, you now have a much clearer picture, a solid foundation for understanding how a normal cell makes that journey to becoming a neoplasm.
Definitely. And you left us with that question about chronic inflammation.
Yes. Why do conditions like ulcerative colitis or chronic hepatitis increase cancer risk? The answer lies squarely in that promotion stage we discussed. Chronic inflammation means constant tissue damage and repair. That relentless cell turnover, the constant regeneration, acts as a powerful promoter. It provides endless opportunities for cells with initial mutations, initiated cells, to proliferate, expand their numbers, and crucially, acquire more mutations during DNA replication. So the inflammation itself might not be the initiator, but it creates the perfect storm for initiated cells to progress towards malignancy. It fans the flames.
Got it. The constant rebuilding creates more chances for errors and helps the damaged cells take over. Makes perfect sense. Well, this has been a fascinating deep dive. For you, our future medical professionals, grasping this complex cellular story isn't just academic. It's fundamental to recognizing risks in your patients, understanding prevention strategies, appreciating why treatments work, and ultimately tackling this disease. So here's a final thought to leave you with. Considering this intricate dance we've discussed, the interplay between these external triggers, the internal genetic controls like accelerators and brakes, the stepwise progression, what part of this entire process do you think holds the most promise for developing new, more effective cancer therapies in the coming decade? Where should we be focusing our efforts? Something to ponder.